Okay, thanks for coming along to the poster session, everyone. I'm Tristan Stark. I work here with Barbara and Malgorzata, and I'll be chairing this session. So we'll get straight into it. First up, we have Fred Jaya, who's sorry, who's made a poster about evaluation of recombination detection methods for viral sequence analysis. Sorry about that, I misplaced my notes. So if you want to share your poster, Fred, and we can take any questions about it.
Sure. Thanks, Tristan.
And you might like to just give us a little short rundown of what you're showing.
Yeah, absolutely. How long do we have to talk about it?
5 minutes.
5 minutes? Cool. Great. Thanks very much. Well, hi everyone. I'm Fred Jaya, presenting from the very sunny Sydney today. It's forecasted about 27, 28, so happy days. And I'm very excited to be able to present my master's work with everyone today. And it's on an evaluation of recombination detection methods for analysis of viral sequences. So firstly, why do we need to detect recombination? What are recombination detection methods used for? So it's quite well known that recombination, when unaccounted for, can mislead some evolutionary analyses. And for example, in phylogenetics, unaccounted recombination can lead to distortions in branch lengths and topologies. So recombination detection methods are used to find regions in a sequence alignment where recombination isn't occurring, and they are then subsequently used for analysis. For example, if we're interested in how a viral population has spread through time, or if we're interested in the origins of a viral outbreak. However, there are many recombination detection methods that have been developed over the past 20 years. And previous benchmarking studies have aimed to provide guidelines for selecting which method is suitable for what sort of data set. However, these benchmarking studies fall short in that they don't really provide any information on which ones are suitable for analysis of more recent sequencing data. And one problem of recent sequencing data for recombination detection is that we've got a lot of viral sequencing data sets that are quite large. And for example, some recent data includes a within-host population of hepatitis C virus deep sequencing, which has about 6,000 sequences that require analysis. And more topically, over now 200,000 global coronavirus sequences that require analysis. And also, some long-read sequences at the moment, for example, PacBioSQL to Hi-Fi Biochemistry is able to output around 4 million reads that cover a lot of RNA virus whole genomes. So what I did for my project is that I benchmarked 5 recombination detection methods. And to do this, we simulated a within-host population of hepatitis C virus. And our first key finding of when we analyzed our simulated data is that sequence diversity has a massive impact on how recombination is actually detected. And to provide some examples of that, firstly, we find that analysis of sequence diversity can be problematic. Analysis of sequences with low diversity can be problematic. And here, so the 5 detection methods that we looked at include FIPAC using the profile option. So if David is in the crowd listening somewhere, I would love some feedback on what you think about this, and I'm gladly to hear you tell me that I'm wrong. But so firstly, in FIPAC profile, we found a lot of false positive detections when sequences were quite similar. And in 2 other methods, 3seq and GeneConf, some analyses could not be run due to a lack of polymorphic sites. And secondly, with simulated data, we found that methods might not be able to recover the simulated recombination events represented by the abundance of red and orange here. And that might be, we think that might be an artifact of our simulations. Maybe our simulations aren't, we're not simulating as much recombination as we expect. or maybe that a within host population has a weaker recombination signal. And lastly, we found behavior of each method that was specific to the method. And for example, GeneConf here at a very particular sequence diversity, it started detecting false positives, and we think that's attributed to its BLAST-like algorithm being sensitive to similar sequences after monomorphic sites are removed. We also had a look at empirical data. We looked at two empirical data sets of bovine RNA viruses, which is a beta coronavirus, and a bovine viral diarrhea virus. And those results supported what we found in the simulation findings, where sequence diversity had an effect on how recombination is being detected. And we find here that 3seq GeneConf and GMOs were restricted to analyze recombination between sequences within a limited range of pairwise distance. So 3seq and GeneConf are more suited for sequences that are slightly more divergent, whereas GMOs less so. And lastly, we looked at how scalable these methods were. Yep.
Can I just interrupt you?
Sure.
We just need to move on, I think, because I might have taken a couple of minutes getting you introduced, but we'll have a little time perhaps for questions at the end. Sorry to cut you short, but we'll have...
Let him finish it, like a final couple of seconds.
Yeah, sorry.
Yep. Yeah, that's all good.
You can have another minute.
Yeah, I'll briefly summarize. And yeah, so to briefly sum up, we looked at the scalability, and the methods that were scalable may not be suitable for analysis of viral data that is highly similar, so UCHIME and GMOs, whereas the ones that may be a bit more robust, such as 3seq and GeneConf, were not scalable. And with that, I'll finish there and take any questions later then.
Thanks, Fred. Sorry to interrupt you there.
Will we have questions now, or are you saving for the end?
Yeah, maybe that's more organic if we can do questions in between.
Yeah.
I just had thanks for interesting talk over there. I was just wondering about like the fail category that you have in your figure there. I was sort of thinking if sequences really are just pretty much identical, would it be, I mean, what you'd probably want a method to do is to say, I don't know, maybe they're recombinant or maybe they're not. So by fail there, is it just that the method sort of saying like just not making a call one way or the other, and is that necessarily a bad thing, I guess?
Yeah, no, I think you're absolutely correct. Like if you're comparing identical sequences, then there is absolutely no recombination signal. But in the case of fail here, when you try to process an input faster alignment, for example, it doesn't run. It doesn't run because there's not enough informative sites. But then that poses an interesting biological question where we expect homologous recombination in viruses to occur more frequently in regions where it's more identical. So computationally, it's a bit difficult to find that.
I've got a quick question for you. That was a great talk, Fred. Why did you choose those particular recombination algorithms? Why not others? And do you look at RDP4, for example? And...
Oh, great question. Yeah, great question. So we were looking at ones that were potentially scalable. So the big criteria for that was that you can incorporate that into a pipeline like Nextflow or SnakeMake. So Unix friendly, not a GUI. So RDP4 is a GUI. I tried running it with Wine, and it's not gonna lie, it's a nightmare. But 3seq and GeneConf are included in, they're two of, I think, the 7 algorithms in RDP4. And I guess all of the 5 methods here, they detect recombination breakpoints. So for example, the base 5-pack looks, I think, for a recombination signal across the entire alignment, but we're interested in finding the method that can pinpoint the location of the recombination breakpoint.
That makes sense. Thank you.
All right, we'll leave it there for the questions for now, and if you have any further questions for Fred, you can forward them to him in the chat or talk later. So our next poster presenter is Nick Fisk, who's going to be talking to us about phylogenetic experimental design via the signal noise framework. Nick, if you could share your
I have to. Jaya's
screen
going to
to
have to
show us
stop.
your post.
Fred's going to have to stop for me to do that.
Yep.
Oh yeah, could you stop sharing yours, please, Fred? Sorry.
Oh, there we go. Great, great talk, Fred. Screen 1. Cool. Can everybody see? Okay.
Yep,
All right.
we've got it.
So I'm here to talk a little bit about phylogenetic experimental design. A little background about me is that I'm at Yale. I'm in the program for computational biology, bioinformatics, but I largely work on cancer biology problems, and I work pretty close with individual doctors and physician researchers there. And so my exposure to sort of phylogenetics and evolution has been mostly through my mentor, Dr. Jeffrey Townsend, who himself, at the time of me joining the lab, shifted very, very much into this cancer work, but in sort of his old life, really worked on phylogenetic, sort of phylogenetic information and specifically sort of quartet-based methods for figuring out phylogenetic signal, phylogenetic noise. So in the sort of 5% of time I get to dedicate to this, I really enjoy it. We kind of wanted to lift some of the ideas from the sort of medical community. When you design a clinical trial, right, one of the things you have to do sort of prospectively is do a power analysis, right? Figuring out exactly what you like, what you need to sample, who you need to sample, and how you need to do it in order for your study to get approved. So using a sort of signal noise framework that's been sort of long established, I helped code up this PhyInformer package. It's on CRAN if you want to look at it, and it was a sort of robust way of data exploration. It would let you do sort of analytically and sort of empirically estimate of quartet internode resolution probabilities. So you could decompose your tree into quartets, and using the sort of site rates and everything, you can get the probability that you will, given your data, resolve your specific quartet correctly. Of course, getting the idea of being a homoplazy, getting a sort of incorrect inference, or a polytomy being sort of not enough power. And so what we wanted to do is do the same sort of prospective power analysis for evolutionary studies. So when someone goes out into the field to sample things, it's one of those things that's getting harder and harder to know what genes you should even include in your studies. Dornsberg, the other collaborator here, and Dornberg and Townsend, they put out a paper recently about the optimal character sampling for that, for sort of rates across trees. But we wanted ability for someone to basically put in a matrix of a gene and taxa. I guess this would be good here. We have a data matrix where these black boxes represent what you've already sampled, a data mask. So these X's are sort of things that are inaccessible, either because there's complications in the biology, or you literally, like, that organism's, like, you can't just go easily sample its blood or something. Think about like Galapagos species or something. And then you have two trees, a sort of guide tree and an empirical tree. And the idea is that you can do this, I guess one of the advances here that we're describing in a paper that's forthcoming is using Shannon information to calculate probabilities of resolution using any set of character rates, which is the idea that instead of decomposing your tree, you can actually collapse it. So you can see here a phylogeny, and I've actually, instead of decomposing it, I've actually used mutual information theory to collapse it about this internode into a quartet, preserving as much information via sort of mutual information entropy results. And the idea is to sort of iteratively do that using the sort of weaker data in this empirical tree onto the big data here. And what you'll ultimately get is a schema, a sampling schema of sort of gene taxon sampling in a ranked order. And you can keep running this iteratively until you reach the threshold probability that you want. So if you want to have a probability of 0.85 to resolve your specific phylogenetic hypothesis, it'll do so. So you don't waste your sort of sampling time and power and even introducing more noise by including things that aren't going to help you answer your sort of specific question of interest. So a lot of this stuff is already on ultimate signal noise. It's a little rough at the moment. It's not ready for like lazy loading by like dev tools or remotes. but I'm getting there. And since Fly Informer was on CRAN, I'm going to try to get it back on there, but it looks like we might have to put it on Bioconductor because I want to include IQ tree and their rate estimation in case users don't know how to infer their own sort of site rates. So overall, that's my poster, and I'd be happy to take any questions.
Thanks very much, Nick. Do we have any questions?
Quick question.
Yeah, you go.
Tristan, just a quick one. I don't know whether I missed it, but Nick, I love the idea. How long does it take to converge? How does that scale with N?
So there's sort of two answers to that. It's actually not as slow as I thought it would be, because ultimately you're relying on the fact that your user has already sort of inferred one, an empirical tree and a guide tree. Though I do have ways of doing it using IQ tree again bundled in here. But one of the things that speeds it up a lot is a lot of site rate estimations will use like a gamma parameter. And that gamma parameter is like, it repeats, right? You only have like 4, 5, 6, usually no more than like 10. And so a lot of the calculations are redundant as you're doing the search, right? So if you go to the GitHub, there's a little animation for it. But if you have sort of very close or at times, a lot of the calculations as you're searching through the matrix is the same. Like there's a lot of redundant calculations. So even though if you did every calculation, it'd be really hard, you can sort of just throw that in a hash map and look it up real quick for the processes that are already done.
Cool, thank you.
Okay, thanks, Nick. I think we'll move along, and any further questions you can direct to Nick through the chat or a little bit later on. So our next poster presenter is Albert Sowong-Sono, and he's going to be talking about the shape of phylogenies under phase-type distributed times to speciation and extinction. Yep, you've got your poster up, so I'll hand it over to you, Albert.
Yep, thanks, Tristan, and hello, everyone. Thank you for having me in this session. So this work is part of my research for my PhD studies, and some results here are actually in the paper that we are currently working on. So in this poster, we are going to talk about the shapes of phylogenetic trees under probability distributions known as phase-type distributions, and we impose these distributions to two macroevolutionary processes on the phylogenetic tree. So we have a speciation process, and also we have extinction process. And by shapes, I mean that we are going to talk about the tree balance and also the distributions of branch lengths on phylogenetic tree. So maybe let's start by talking a little bit about the phase-type distribution. So phase-type distribution is basically a distribution that models times until you reach the absorbing state in a continuous time Markov chain. So you have n transient state, and also you have your absorbing state, and it is defined by two parameters. So you have your alpha parameter, which basically records the initial probability of starting in any one of the states, transient states. But for the Coxian one, the structure is that you are only allowed to start from the first state. And also you have this rate matrix Q that records the transition rates going between the transient states. So biologically, you can think of your species of going through different phases in their life until they either go extinct or they speciate. Right, so that's about the Coxian phase-type distributions. And then the way we do it is that if you look at this figure here, so each branch length, we assume that we have independence of branch length, and we can do that if we assume like symmetric speciation mode. So in symmetric speciation mode is that we're following a speciation events, both lineages, the children lineages, they don't inherit the parent's age. So you can draw the time independently from the phase-type distributions. And so to do that is, are we going to simulate, say, 600 trees, each with 100 living tips, and then we do in two simulation. So the first part is where your time to speciation events are drawn from a phase type distribution with some parameter choices, while the time to extinctions are drawn from an exponential distributions, while you do the reverse for the second simulations. Now the times to extinctions are drawn from phase type, but your times to speciation are drawn from exponential. And then we're going to see the changes on the tree balance or the distribution of the branch length. And to do that, we need to quantify them. So for the branch length, we do that by computing these beta statistics. So if you have, for example, your beta negative, that means that you have unbalanced trees. That's like this figure on the right. But if your beta is positive, you have like balanced tree, like this one on the left. And then you have this branch length quantified by this statistic known as gamma statistics. So that indicates like whether you have a slowdown in diversifications or you have an increasing diversifications in your phylogenetic tree. So these are the simulations. So the figure on the left, that's where we have the time to speciation drawn from phase type, while time to extinction follows exponential. And we can see some like slight changes on the beta statistic, but that is not the case for the gamma statistic for the branch length. But when we do for the extinction process that follows phase type distributions, we see some changes on the branch length, but not for the beta statistic. But the figure isn't plotted here because there isn't enough space. So so that's one of the results. And the other result, if you look here, you just these 4 different pairs of X and Y. You can see that you have beta that is negative, but you also have beta that is positive. So it's possible, like with more parameter choices from phase type distributions, you can basically generate trees with different balance level. I think that's all. Thank you.
Thanks, Albert. And we'll take some questions for Albert if anyone has any questions. So one thing I guess then I wonder is, have you looked at any other sort of types of phase type distribution besides the Coxian to look at the balances that result from other models?
Yeah, so I think like we try with the Coxian phase type distribution. That's because like there are some results that basically states that if you have a cyclic phase type distribution, so in this case you have a change that moving forward, you can represent that with a Coxian phase type distribution. So that's why we start with the Coxian phase type distribution to study the shapes from phylogenetic trees.
Thanks, Albert. Any other questions for Albert before we move on? Let's thank Albert again.
We'll mention if you're interested in Albert's topic, he's giving a talk tomorrow in the final session.
Yeah, there'll be more stuff on that. And
our last poster presenter for today, who we've heard from earlier, is Songhwan Jun. And he's going to be talking about generalized pruning. So if you could share your poster with us, Song.
Yep. Can you see it?
Yes.
Yours.
Okay. Thank you for the introduction. So I guess I'll go into a little bit more details on this subsplit business and talk about this generalized pruning algorithm. So although my talk itself doesn't have variational components to it, but basically the key idea underlying the variation of phylogenetics for topologies is this idea of subsplit representation of a tree. So a tree can be decomposed into smaller building blocks. So here we have a root split. So you basically choose how you're going to split all of the taxa. So in this case, we've split it A from B, C, D. And then you can further subdivide this B, C, D into B splitting off from CD, and so on. So this is one way to view a tree, and we call these ones so parent subsplit and child subsplit. And the nice thing about this decomposition is that you can specify a support set over trees by conditional probability table or conditional probability distributions. So for example, in this table, I've shown we have two choices. A, B, C, D could be split in two ways, as shown here. So with probability 0.3, you can get A splitting off from B, C, D. And with 0.7, you get A, B split from C, D. So what this means is that you can compute the probability of a given tree by just multiplying these conditional probabilities. So for example, this tree has probability of 0.27 because 0.3 for choosing the loose split and 0.9 for splitting BCD as such, so B from CD. So this subsplit representation is nice also for dynamic evaluation of tree probability, which we use in generalized pruning algorithm. And that's that you can easily compute the probability of a subtree, which is just by multiplying the probabilities of the parent-child subsplits that show up in the tree. And you can also evaluate the probability of partially specified trees. So here we have a tree where what happens below this node here is not really specified. And we can evaluate this tree probability the same way we did for the full tree. And in this paper, Zhang and Madsen, they proposed this idea. They show that this representation seems to capture the posterior distribution sufficiently well when compared to MCMC. So here, the BIMCO and RWS are two ways of fitting a variational approximation. And they run Mr. Bayes for 2 million iterations, and they noticed that the variational approximation parameterized by subsplits achieve much better, lower KL divergence. So we're going to use this representation to build a new algorithm. So my generalized pruning algorithm is not variational inference. It's actually, we're trying to eliminate trees from parameter estimation so that we reduce uncertainty. So we built this graph called ordered subsplit graph. And this is basically a graph built on the subsplits. So this graph encapsulates all of the trees that are spanned, that are in the support specified by this conditional probability table. And basically what we're going to do is we're going to pass the messages up the graph. So here blue arrows show the messages direction. So we have messages coming into S from U and W. And then it does computation to combine the messages from the children and then combines it and passes it up to the parent. And this is basically similar to the way Felsenstein pruning algorithm works. You have messages coming up from the left and right child. You combine it using element-wise multiplication and passing it up to the parent. The difference here is that we actually each of these nodes can have multiple children instead of just left and right child. So we have this summation to account for that. And we also have a message going down the tree. So it combines the message coming from the parent with the message coming from one of the children, and then passes down to the other remaining children. So doing this lets us compute marginal likelihood for single site is exact. And combining those exact single site marginal likelihood gives us composite likelihood, which ignores the dependence across the sites. But if you have large number of sites, this could be a good proxy for using it in estimation purposes. And we used this estimation procedure and show that they seems to be doing a good job of estimating on the probability, the conditional probability table that I showed in the previously, when compared to other methods used in Zhang and Madsen. Yeah, that's it. Thank you.
Thanks, Song. Do we have any questions? Barbara?
I guess, I mean, obviously you think about this, applying this more in a sort of a Bayesian setting, but I wonder, like, does it open up the possibility of getting an effective branch and bound algorithm for maximum likelihood in a similar way as, there have been branch and bound algorithms to exactly solve parsimony for sort of medium-sized problems?
Yeah, I personally haven't thought about that direction of research. But one possible one avenue of research that at least some people in my group are thinking about is so using this to sort of expand the space of trees. So that's maybe where branch and bound could come in and could be useful. So here we assume that support of these conditional probability tables are given to us, but like ideally we would like to maybe start with a small set given maybe from initial run of I don't know, like bootstrapping or something like that. But then we would like to expand the support in some natural way. And maybe that's where some of the maximum likelihood, the parsimony-based ideas could be useful. Yeah, does that answer your question, or did I?
Yeah, no, thanks, yeah. Thinking aloud.
Thank you.
Any other questions before we wrap up? No? In that case, let's thank all of our presenters from this session again. And we'll see those of you for whom it's an appropriate time for this evening's session. So thanks again.